everyone! In this PowerPoint presentation, I will go over the different types of tissues by giving examples of each of them. Let's get started. First, we have a list of terminology which will be used for the assignment, so make sure to take a look at it when completing your assignments. Let's first go over the epithelial tissue. Let's start first with the simple squamous epithelium. Simple means that this epithelium is made of one cell layer, and squamous means that the cells are flat. Sometimes it's going to be difficult to visualize the whole cell with its plasma membrane. However, with this staining, you will be able to visualize the nucleus. If the nucleus is flat, it means that the cell is flat. Therefore, on the image to the right, which is representing the cornea of the eye, notice how these nuclei are flat and they are spread into one layer. Therefore, this means that this epithelium is a simple squamous epithelium. Also note that the basement membrane, which is located under the epithelium, is pretty thick on the cornea, as it is this dark pink line right under the flattened cell nuclei. And under the basement membrane, what do we have? Well, we have the presence of the connective tissue. In this case, notice how all these collagen fibers are going to be oriented parallel to each other and how they are densely packed. This means that this connective tissue is going to be a representation of the dense fibrous connective tissue. But we will talk more about connective tissue later on in this presentation. To the left, we have a kidney tissue. These large round structures are going to be called the renal corpuscle, also known as the Bowman's capsule. Inside the capsule, we find a large quantity of blood vessels all mingled together. These specialized capillaries will form what we call the glomerulus, of which we will learn when we cover the urinary system. So for now, what I want you to notice is that the glomerulus, which again are all these grouped blood vessels, are going to be wrapped by this thin layer of epithelium. Notice how the cells have a flat nuclei and are organized into one cell layer. This means again that it is a representation of a simple squamous epithelium. Here we have a sample of a bile duct tissue. And bile ducts are going to be present inside the liver. This cross-section of the bile duct contains many blood vessels that are going to be located in the surrounding connective tissue. We have one here on this image and a closer look on this other image to the left. Notice all these bright red cells inside of the blood vessels. Well, these are going to be the erythrocytes or red blood cells. Makes sense, right? The red cells are called red blood cells. However, notice that the dark blue nuclei around the blood vessels are flat and arranged into one layer. Therefore, blood vessels also have a simple squamous epithelium. We can go even a little further by saying that the epithelia that line blood vessels, the heart, and the lymphatic vessels are also known as endothelium. This means that even though this is a representation of a single squamous epithelium, it can also be called an endothelium. And the cells that form this endothelium are termed endothelial cells. Therefore, if this term is used, you know that it is going to be referring to blood vessels specifically. So keep that in mind. Next, we will cover the simple columnar epithelium. This means that it will be made of one cell layer, as it is termed simple, and the cells now will be rectangular, tall cells, meaning that the height is more than the width of the cell. Notice that this image to the right, representing the wall of the bile duct, how these cells are going to be tall, and the nucleus is round and located closer to the connective tissue. Since we are able to see only one nucleus in a row on this side of the tissue, this means that this is a simple columnar epithelium. Sometimes it can be a little bit confusing. Notice how on the left side of this tissue, it seems that there are several cell layers, as we can locate several nuclei spread out in several layers. But this is going to be due to the angle that the side of the tissue was sectioned. Therefore, keep this in mind and try not to get confused. The image to the left is going to be a sample of the pancreas. The ducts usually have a simple cuboidal epithelium as seen in these examples on the right of the specimen. Notice how these cells look cuboidal or square. When you compare these cuboidal cells with the cells on the left of the specimen, notice how these on the left appear more rectangular or columnar. Therefore, always make sure to look at the shape of the cell to be able to identify the different types of tissues. On this next slide, we have more examples of simple columnar. However, here we are adding a different type of cell. See these big round cells that look clear? We can see them in all of these samples. These cells are what we call goblet cells. Notice how they are not in a row. They actually appeared scattered throughout the tissue. And the function of these cells is to secrete mucus. Also notice how these goblet cells are located near the apical surface, or the top part of the tissue. And this makes sense because since this cell's function is to secrete mucus, it makes more sense that it would be located further up on the tissue, closer to the lumen, or the outside where the secretion will be released. And how do we know this? Well, look how the nuclei of the epithelial cells are located opposite to the goblet cells. And we know that the nuclei are usually located on the basal surface of the cell. In other words, the surface that is going to be closer to the basement membrane that will sit between the epithelium and the connective tissue. In addition, we can see how these cells look clear compared to the other epithelial cells in this tissue, and this is going to be due to the mucus being extracted during the preparation of this specimen. And what specimen are these? Well, you should be thinking, where would you want to have mucus secretion? One location would be in the digestive system. Therefore, these two images over here are representing the ileum, and this other image is representing the jejunum. Both are going to be part of the small intestine of the digestive system. And let's not forget that because these epithelial cells are taller than they are wider, and the nucleus is arranged in one cell layer, this epithelial tissue is a representation of the simple columnar epithelium. Moving on to what we call the pseudostratified columnar epithelium, where pseudo means false. Therefore, this means that this tissue is a false stratified. If it is false stratified, it actually means that this tissue is actually a simple tissue made up of one cell layer. However, these epithelial cells, they're packed so close to each other that when the tissue is sectioned, it looks like there are several cell layers because, as we can see on this image, the nuclei are going to be located at several levels of the tissue, giving this impression that there are several layers of cell. But since this tissue is a simple epithelial tissue, it means that the basal part of all of these cells are actually attached to the basement membrane. The other thing that we notice on this tissue is that on the apical part of the cells, we can see a thin layer on top of the cells. This thin layer is representing the cilia or hair-like structures that are present in this tissue. And what sample of tissue are these? These two are from the trachea, and this one on the far right is a sample from the glottis, meaning that they're all from the respiratory system. And the respiratory system is characterized for mainly having pseudostratified columnar epithelium. Now, because we have the presence of cilia, we can add this word to the classification of this tissue. Therefore, we can call it a pseudostratified columnar ciliated epithelium. In the simple cuboidal epithelium, what do you think we should be looking for? Well, simple means it is one cell layer, and cuboidal is referring to the cell shape. In this case, it will look like a cube, therefore a square. So we're looking for one cell layer of squared cells. Both of these samples are coming from the kidney, and remember the structure here? We have already seen this structure previously. What is it called again? If you said the renal corpuscle, you are correct. It can also be called the Bowman's capsule. Well, the area around the renal corpuscle usually contains many cross sections of tubules with a simple cuboidal epithelium. We can see these right over here, forming a cluster of tubules. And on this image to the right, it seems like the only thing being represented are these tubules. Notice how they form a circle of cells that look cuboidal and that are all arranged in one layer. And this space in the middle is called the lumen of the tubule, where there will always be a substance passing through them. In case of the kidney, this is probably the filtrate that will later give rise to the urine that is then released through the body when you urinate or pee. Here we have a few more examples of simple cuboidal. The tissue on the left is a sample of an ovary. Notice how these outer cells forming the epithelium are arranged in one single cell layer, and the cells are square. The image to the right is representing the pancreas. Again, squared cells in one cell layer. Previously, we have covered all of the simple epithelia. Now we will move on to the stratified epithelia, meaning that the cells are going to be arranged forming several layers. Let's start with the stratified squamous epithelium. The typical example of this type of tissue is the esophagus. All of this is representing the epithelium. Therefore, we can clearly see that there are several cells going from the basal side of the tissue all the way to the surface. Notice how the cell nuclei of the basal side look round. However, as the cells reach the surface, notice how the cell nuclei appear flatter. Therefore, for us to classify a stratified epithelia, we must look at the last cell layer, the ones that are closer to the surface. In this case, since they look flat, we classify this tissue as squamous. Thus, we can say that it's stratified squamous epithelium. And why would you need several cell layers? Well, as food passes through the esophagus, if you do not chew your food well enough, it might damage the surface. And if it was only made of one cell layer, then this damage would probably affect the underlying connective tissue, and this might cause some sort of infection or injury. Therefore, the importance of having several cell layers would be for protection. This also means that these cells that are removed during swallowing are going to be continuously replaced by division of the cells that are going to be present in the basal layer. Here is another example of stratified squamous epithelium, which is the skin. There are two types of skin, thick skin and thin skin. Thick skin, which is represented here, only occurs on the palm of your hands and sole of your feet, whereas thin skin occurs on all other body parts. This slide shows, like I said, the thick skin. So the thick skin is also covered by what we call a stratified squamous keratinized epithelium. What does keratinized term mean? Well, since the skin is going to be exposed to air, it is keratinized to protect the surface from the abrasion and is lubricated by glycolipids to protect it from dehydration. This outer layer is representing the keratin layer, and the layer below is where the stratified epithelia will be located. We will be able to see these cells in more details on the next slide. However, still on this slide, I want you to notice the thickness of the keratin layer, and we will compare it to the thin skin on the next slide. So again, this is the thin skin. Notice how the keratin layer is not as thick as the one found in thick skin. So this is why this is called thin skin and the other one is called thick skin, mainly because of the difference in the width of the keratin layer. But of course, the more keratin you have, it means that it serves more for protection. So here is the keratin layer, and below it we see several cell layers. Also notice how the nuclei of the apical layer look flatter than the cells located in the basal layer. Therefore, it is a stratified squamous epithelium. And because there is the presence of keratin, as seen on both images, we add the term keratinized. Therefore, stratified squamous keratinized epithelium. We can't really see the basement membrane as it is too thin to be able to identify on these samples. Next, we have the stratified cuboidal epithelium. This type of epithelium has a limited distribution, and it is going to be most often found in large ducts of the exocrine glands. Typically, it will have only 2 layers of cuboidal cells, but since it has more than 1 layer, it is already termed stratified and not simple. Notice over here 2 nuclei, one on top of the other, and on this duct as well, 2 cell layers. This tissue is from an esophagus and will be located within the connective tissue of the esophagus. Moving on to the last type of epithelial tissue, which is the transitional epithelium. This type of epithelium is going to be found in the lower urinary tract, like for example the ureter and the urinary bladder. It is called transitional because it rapidly adapts. And what does this mean? Let's think, for example, about your urinary bladder. When your urinary bladder is full of urine, the tissue will need to stretch to be able to accommodate the urine that is being stored in the urinary bladder. Makes sense, right? Therefore, the cells are going to stretch with the tissue, right? In this case, this transitional epithelium will be called extended transitional epithelium. Once your bladder is full, you will need to urinate to empty it out, and therefore the tissue will return to its original shape. And this tissue will then be called relaxed transitional epithelium. And as we analyze the 2 slides on the images of this PowerPoint, notice how the apical cells look rounded compared to this other slide where the surface looks flat. This means that this tissue is relaxed and this other tissue is stretched. Therefore, this one over here would be your example of relaxed transitional epithelium, and this one would be your extended transitional epithelium. Now that we're done with the epithelial tissue, let's move on to muscle tissue. There are 3 types of muscle tissue: skeletal, cardiac, and smooth. Both the skeletal and the cardiac are not smooth. What does this mean? It means that they will have what we call striations, or these lines that run opposite to the length of the tissue. Let's now look at this characteristic on these slides, which are both a longitudinal section of muscle tissue. This slide on the left is an example of a skeletal tissue, and the one on the right is an example of the cardiac tissue. Notice the cell running parallel to each other on both slides. Also notice these lines running transverse to the length of the cells. We can clearly see them over here on the skeletal muscle tissue. Since we have these 2 types of muscle tissue on the slide, let's compare the 2 so that you're able to differentiate them. The first thing is that on the skeletal muscle tissue, the cell nuclei are located on the periphery of the cell. See how they are on the outside of the cells, while the nuclei of the cells in the cardiac muscle tissue are located in the middle of the cell. We can see one here, another one here. Also, with regards to the nuclei, notice how the skeletal cells have several nuclei on the periphery, while the cardiac cells have only one nuclei that, like we said, are located centrally. The other difference is with regards to what we call the branching of the cells or splitting of the cells. Notice how the skeletal cells are long and run straight. In contrast, the cardiac cells split or branch. See how the cells split over here, for example? Therefore, we can say that the skeletal muscle cells do not branch, while the cardiac muscle cells do branch. There is one last structure that is present in cardiac muscle tissue and not in skeletal muscle tissue, and this is the intercalated disc. One can be seen right over here, another one here, and we will learn more about these when we cover the cardiovascular system. However, we can say that these will be important to hold all these cardiac cells together when the heart is beating. Remember that the previous slide, the tissue was sectioned longitudinally. Here we have two cross sections of the skeletal muscle tissue on the left and cardiac muscle tissue on the right. How do I know this? Well, notice how on this image to the left, there are several nuclei located on the periphery of the muscle cell. While this image on the right, there is one nucleus that is located centrally. And this is a great example, isn't it, for you to be able to differentiate the two types of striated muscle. On this slide, we will go over the smooth muscle. Notice how we don't have any of the striations on the smooth muscle. And this is why it is called smooth. It's pretty simple, right? And this image on the left has an example of a longitudinal section on the bottom and a cross section on top. And I have zoomed in here to show you in more details. Notice how in the longitudinal section the fibers run parallel to each other, while in the cross section they don't. That was it for muscle tissue. Now we're moving on to nervous tissue. So let's talk about the different types of staining first in the next slide. As we can see here, we have 3 slides that have a purple staining, and this staining is called crescent violet. Crescent violet stains the nuclei of both the neuronal and the non-neuronal cells. The non-neuronal cells are going to be what we call the glial cells. Therefore, these big purple cells located in a cross-section of the spinal cord are the neurons, and these smaller cells are the glial cells that will have a function of supporting the neurons. This image shows a sagittal section of a rat brain showing several neurons and smaller glial cells as well. And this other image is a cross-section of the dorsal root ganglion. These large purple cells are going to be the neurons. We're able to see the nucleus inside of the cell as well. And these smaller purple dots are stained nuclei from the glial cells that are present, like I mentioned, in the dorsal root ganglion. This other slide is stained using what we call a Golgi method. And in this method, it stains randomly a limited number of neurons and their processes, which is everything that sticks out from the cell body, therefore the axon and the dendrites. This will allow the morphology of individual neurons to be examined. Here, for example, would be the cell body of the neuron. These would be the dendrites, and this long process would be the axon of a sagittal section of rat brain. In this staining, the non-neuronal cells of the nervous system do not stain, so the glial cells cannot be seen. And that was it for the nervous tissue. Now we're going to be covering the connective tissue. The connective tissue is located right below the epithelial tissue and the basement membrane. There are 2 main types of connective tissue. the dense and the loose. Where the dense connective tissue is reinforced by bundles of fibers that will provide a tensile strength, also elasticity and protection. And in loose connective tissue, the fibers are loosely organized, leaving large spaces in between them. In addition, the dense is divided into regular and irregular, where the regular means that the fibers within the tissue will be well organized, while the fibers in the irregular tissue will be, well, irregular, right? And the name is telling you exactly what it is. So let's identify those images. Let's start with this one, which I think is the easiest of them all. Notice how there are several fibers and they are all well organized. Therefore, it makes sense to identify this connective tissue as the dense, regular connective tissue. This image on top also has several fibers. However, these fibers are not as organized as they are running in several directions. This means that this connective tissue is the dense, irregular connective tissue. Moving on to this image on the top left, notice how we can see some fibers spread out in several directions with several cells in between them. And these fibers are collagen and elastic fibers, while the cells are the fibroblasts that will produce the collagen and elastin to form these fibers. This tissue is what we would call loose connective tissue, also known as areolar connective tissue, as areolar means space, and we can see how there is plenty of space between these dark lines that are representing the fibers. Lastly, this image on the bottom is also showing an example of loose connective tissue. Notice all these fibers with several space between them. In addition, we are able to see all these cells that are empty. These are adipocytes or fat cells, and they are the largest component of the loose connective tissue. These fat cells, they do appear empty because the lipids are actually extracted during the tissue preparation. And lastly, here's an example of fluid connective tissue. This is a blood sample showing a few lymphocytes in purple, and all of these smaller red cells are erythrocytes, or like I said previously, the red blood cells. And with this, we summarize all the different types of tissues.